Welcome to the 12-2 podcast, and this one's on chromosomes and on DNA replication. And this is where we find out how do we copy these things. So we're talk about prokaryotic cells. Remember, they do not have a nucleus, so the DNA is located in the cytoplasm. And most of them have a single DNA molecule with all the material on it, all the genetic material on it. And here's a picture of one. There's the chromosome in E. coli bacteria, and you can see the bases look the same. And so eukaryotes have about 1,000 times the amount the DNA as prokaryotes do. And here's here's a prokaryote versus a eukaryote, and the DNA is located in the cell nucleus, inside chromosomes, and the number of chromosomes varies widely from one species to the next. There's a worm that I think has 50 chromosomes or something like that, whereas humans only have 46. So the complexity of the organism does not correspond with the number of chromosomes. So, and DNA molecules are really long, and here's a picture of a E. coli cell that's been opened up, and the DNA has spilled out of it, and you can see it's just everywhere. And you're trying to fit this enormously long piece of molecule inside a very small area. And the analogy I always like is it would be trying to fit 300 meters of rope into a backpack. That's pretty tight. So the chromosomes contain DNA and a protein, and tightly packed together, these two, you create chromatin. And the proteins are called histones, and the DNA is coiled around it. So a DNA and a histone molecules form nucleosomes, and the nucleosomes pack together, forming a thick fiber. So here's a look at it. Here's your DNA. Here's the histones. These are proteins. The DNA is coiling around the histones, and when they coil around into these balls here on the histones, it's what we call a nucleosome. And then nucleosomes coil up into coils, and the coils are coiled up into supercoils, and you can see that all these supercoils go around together to make up the chromosome. So what happens during DNA replication? The DNA, the neat thing about the double helix is that the information is on both halves of the DNA molecule. So each side has all the information you need to reconstruct the other half by using base pairing, which we learned about in 12.1. So DNA replication begins at a single point, and then it goes off into two directions. And you can see here some, and on top of that, DNA replication occurs at hundreds of places at once, because it takes a long time to copy all this. So if you see right here, here's what we call fork. This is where here's one copied strand, and here's another copied strand. Here's the original strand here. And you can see here's one side of it copied. Here's the other side copied until it meets around here, and here's the other fork. So this part right here has all been copied, and this is what's yet to be copied here. And here you can see some replications occurring here and also here. So these are called, like I said, replication forks, the sites where they're being copied, right where those pink arrows are. So here's DNA. So we call this process replication, when the DNA copies itself, and that way each cell will have a complete set of DNA, and an accurate set at that. Here you can see, here's what I was talking about, is here's one of the old strands, here's another one of the old strands, and then the new strands are formed using the old strands as a template, and this new strand here corresponds to this old strand here. They're the same, they have the same code to them. So during DNA replication, the DNA molecule separates into two strands, then produces two complementary strands following the rules of base pairing. Each strand of the DNA double helix serves as a template, that's a great word, for the new strand. So you can see here's the original strand. This is what I showed before, the original strand, the new strand building off on it, and here's the replication fork, and This molecule is called DNA polymerase, and this is the one that actually makes, it's an enzyme that attaches the new nucleotides that make the new new strand to the original strand. DNA first to start is unzipped with a molecule called helicase. So it's carried out by enzymes that unzip a molecule of DNA, and this enzyme is called helicase. And then the hydrogen bonds between the base pairs are broken. Remember, they're not very strong bonds, so the helicase is breaking the hydrogen bonds, and the pieces unwind from each other. Then DNA polymerase joins the individual nucleotides to produce a new DNA molecule, and then it actually proofreads it to make sure it got it right. So you can see here's one DNA polymerase, here's the other DNA polymerase, here's helicase right there, unzipping it, and the copying is going in this direction that way and going in that direction that way. On to the quiz.